Thank you, Sarkis. Let me just get to the screen sharing part. All right, I'm going to just do one thing. Where is annotate? Don't see it. All right, I'll start. But so thanks for this great opportunity. Thank you, Sarkis, for the introduction, and thank you, Pramila, for inviting us and for supporting this work. So this is slide 2. Let me go back to slide 1. Oops, it's going the wrong way. So basically, the project that we're funded to do is connecting rare genetic mutations in autism to common biological pathways. And Matt Lally and I will kind of share the responsibility. I'll talk about the past, what we know about rare genetic mutations. And then Matt will talk about his efforts and our efforts to put these rare genetic mutations into common biological pathways. This is supported by the Brain Foundation. We do not yet have data, but Matt and team are moving very quickly, and we expect we're actually on schedule to finish everything as proposed. So just taking kind of a long view of rare genetic mutations in autism, I'll kind of highlight the work of the Autism Sequencing Consortium, which really started with the last stimulus package in 2009, the American Recovery and Reinvestment Act by these two gentlemen here who signed it off. One retired and one came out of retirement. And a small amount of that stimulus money went to the NIH for some crazy ideas. And one of the crazy ideas was whole exome sequencing, which was a technology that, or whole genome sequencing, that was now really becoming available academically within reasonable prices. This was the first unofficial or official meeting of the ASC in 2010 in New York City. We brought a lot of the funders and main labs who were doing sequencing together, and what was going on in 2010, the papers came out in 2012, was whole exome sequencing was supported by the ARA Act, and several labs, 5 labs, decided to work together with ARA funding and sequence, a huge number of samples in those days, 50 trios a laboratory, and put all the data together and see what we saw. At the same time, the Simons Foundation was doing something pretty similar, funding 3 laboratories to work on the Simons SSC samples. And so over the course of 2012, there were these 4 papers had about 1,000 ASD families and focused on de novo or recent mutations in autism. And collectively, if you analyze it jointly, there were 9 genes that were shown to be contributing to autism in a very significant way, by which I mean, if somebody had a mutation in that gene, it would probably contribute to most of their developmental phenotypes. And based on that, something called the hidden species problem, a simpler version is the birthday problem. But basically, you could estimate with 20,000 genes in the genome, how likely is it that you're going to find 9 genes currently hit by mutations out of the greater number of autism genes that should be in the genome. And at that time, in 2012, the data was pretty consistent and hasn't really changed, that about 1,000 genes in the human genome, 1,000 out of 20,000, can be changed in such a way to contribute to autism risk. So in a certain sense, we all have autism variants, and because 5% of our genes are directly involved in some of the processes that underlie the social manifestations of typical development and atypical development. So the argument basically, and just to make it clear, if there's 1,000 genes, right, and we had finished 1,000 cases in 2012, we're far away from discovering all the genes that we can, right? We're at the very early stages. And so the argument of the ASC was, let's put together all of our data prospectively, not publishing small findings at a time, but try and get all the samples run concurrently, which was a bit of a stretch in those days. It was the first example of its kind, really, of large-scale prospective data sharing and sample sharing. The first paper came out in 2014, and now had 22 genes that were autism genes, overlapping with many other developmental disorders. SHANK3 you just heard about. I won't talk much more about that, but that's a good example of a gene where the mutation contributes to the phenotype: autism, intellectual disability, developmental delay, epilepsy, and obviously also GI abnormalities. The most recent paper from the ASC, from the Autism Sequencing Consortium, came out last year, and now it has 102 genes all across the genome, and there's SHANK3 again, that each one, when mutated in that individual, contributes enormously to the risk for autism. And what's gone on to bioRxiv, so this is kind of what we look at. The total sample size is in the tens of thousands of samples and 12,000 cases in individuals with autism. And each one of these funny looking conglomerate of letters is the name of a gene. Of the 20,000 genes of the genome, here are 25 or so that, you know, a fraction of individuals with autism have mutations in that gene. And here's another group of genes where a smaller fraction have mutations in that gene. There's SHANK3, which you just heard about. And so, you know, obviously, if you are working on SHANK3 as an example, you have a biomarker, for lack of a better word, where you can find people that have a mutation in SHANK3 and study them directly. And you can make a mouse model, as Sarkis told you, that has a mutation in SHANK3 and understand what's going on in that mouse, which reflects what's probably going on in patients with that mutation. So you can take basically a rare disorder or orphan disorder approach to these genes and say, I'm not studying autism, or I am studying autism, but I'm studying the autism that's caused by mutations in SHANK3. The most recent analysis by the ASC now doubles the number of cases, 20,000 cases, more from the ASC, but also many more from the Simons Foundation. And we now have 71 genes at a very, very strict threshold. But at a kind of the typical threshold we tend to use, we now have 250 genes that contribute to autism risk. So remember I said there's about 1,000. So, you know, we're honing in on 25%, which is pretty impressive. And this is the genes listed by basically by evidence of their association. There's SHANK3 you just heard about. ADMP is one of the genes. We study SHANK3, ADMP, FOXP1, a couple other ones. So you can select these genes here and approach them, as I said, as a rare disorder. This study really was, I think, another important step in gene discovery in autism. And it's really been led by a conglomerate of individuals who've done an amazing job of bringing the SPARC data and the ASC data together, and also bringing in other kinds of variation, including copy number variation in the analysis. So that should be online. I think it was submitted to bioRxiv on Friday. It should come live in just a couple of days. So, you know, when I started this business of autism discovery, gene discovery, you know, we thought maybe there'll be a couple of genes. And I remember very well when the paper came out and said that there are going to be 4 or 5 genes underlying autism based on how it looks in families. And the idea was that if we could find the gene, the autism gene, or even those 4 or 5, you know, they would highlight a single pathway, right, out of many one, right? There'd be 4 or 5 genes, strong genes. There'd be one pathway, and we'd know where to go for therapeutics. We can talk about this at length, but first of all, we've gone from 4 genes to now 1,000. And every time we look at those genes, there's clearly not one pathway, right? You know, there are neurons, and Sarkis has talked about some of the indirect effects of a neuronal gene on oligodendrocytes. There's lots of ways where you can disrupt the brain to cause behavioral abnormalities of various kinds, including deficits in social behaviors and the presence of repetitive behaviors. So, you know, what I've already kind of intimated is that a lot of groups scientifically are focusing now on a gene by itself. So out of many, E pluribus multis, right? So instead of saying we treat autism as an entity, we could treat autism as a series of independent disorders, right? So your therapeutic focus is on gene-targeted approaches. You think about treatments that are going to help somebody with a SHANK3 mutation or Phelan-McDermid syndrome. And it may or may not help the next person over. It may, but it also may not. So it's kind of what we do in cancer now, right? And, this is from an epidemiological study in Sweden. If you just take current genetic knowledge, you can actually make a genetic finding in about 28% of individuals with autism. So, there is a path forward for, at least this slice of the pie to say, hey, we can take a rare genetics or orphan disease approach to autism. Of course, there's another larger group that has no genetic diagnosis. So we will make more genetic diagnoses as the number of genes increases. But there's also going to be a significant number that are not going to have a formal mutation that explains much of the phenotype. So this is very kind of where biotech and certainly academia is in autism. They're thinking about a lot of these genes like SHANK3, ADMP, POGZ, SNC2A as rare diseases that can be kind of approached with the orphan drug perspective. And there's a lot of incentives to orphan drug development. It is probably one of the most robust areas of therapeutic growth right now, at least in higher income countries. Some of it has been gene therapy. Some of it's been just remarkable just in the past couple of years. Kids who have certain kinds of developmental abnormalities that will end in their very early death can now get a single injection in the spinal cord and actually, so far, certainly be doing very, very well, right? So the idea of gene therapy or gene-targeted therapy has really come into its own when people are realizing that it can make profound differences in developmental disorders. Obviously, these ones that we're talking about are more severe than autism, but if the treatment or the intervention is safe and the individual has a very difficult trajectory, no language, severe anxiety, severe attentional deficits, low IQ, you begin to think about interventions to kind of to reduce some of those disabilities. Right? So I mentioned that we take this approach of rare variation in autism, and we do a pretty deep dive on 5 genes, SHANK3, ADMP, FOXP1, DDX, which is on the X chromosome, and CHAMP1, which actually comes from another study. We won't go into this, but CHAMP1, ADMP, some of the others actually do form a complex and act together, but others do not, right? And what we try and do with each of those genes is we try and do in vitro work. We try and work in iPSCs or stem cells. We try and look at genetically modified rodents, and we also see patients. So Sarkis was nice enough to talk about some of the genetically modified rodents of SHANK3. We've made a couple, not the one Sarkis talked about, but he mentioned the work of Drew Corelli, which is one of the mice that we've made. But we've also seen a lot of patients with this mutation, and a lot of the phenotype, not only the autism phenotype, but the other behavioral and other phenotypes in Phelan-McDermid syndrome have been carried out by groups like ours that bring in these individuals who really aren't known medically until you bring in a good number of them. And that you also learn something about endpoints for clinical trials. You can develop patient biomarkers, you look at the mice, and you can do stem cell work. And so this idea that maybe, if you look at the mice, as Sarkis said, you'll learn something about the phenotype and interventions that you can then maybe use for a clinical trial. Another possibility is you use in vitro work like stem cells. You try and find agnostic or gene-targeted approaches. You see if they work in your stem cells. You can make your model. You can try them in your mouse, and you can ultimately do clinical trials there as well. So that's a pretty robust direction the field is going. This is one example of in Phelan-McDermid syndrome, actually, where some deficit we saw in the mice, both in the brain function and in motor abnormalities, suggested a certain approach, and we saw some interesting effects of a specific growth hormone, a growth factor in patients with this disorder, which we now submitted a follow-on study. However, as good as all that is, we found 250 genes out of 1,000, and many people will not have a mutation. They might have genetic risk, but not a formal mutation. So this approach is only going to get us so far in the autism spectrum. So the other approach would be not out of many one or out of many, but e pluribus paucis, which means out of many, few. Can we take 100 genes and come down to just 10 pathways? And instead of thinking about treating each gene mutation independently, we think about treating each of those 10 pathways, depending on the individual. And that individual may not even have a mutation. They might just have a deficit in a pathway that other people with autism have as well. And that would be the point of intervention. So it is this pathway targeted approach. So then the question becomes, how do you get from these rare genetic mutations to actually find common biological pathways? And that is really the subject of the funding. And Matt is going to take it from here. So Matt, are you going to share your own screen, or do you want me to just advance?
I can throw mine up, I think.
Okay.
Yeah, so thank you, Dr. Buxbaum, for that fantastic presentation. 2,000 years to develop therapeutics for the existing genes that we've already discovered. And as the rate of gene discovery continues to grow, it's clear that we need new approaches to try to come up with treatments that can work for many of the genes at once. But before we can do that, we need to establish a system that would allow us to study many ASD risk genes at once so we can discover their shared pathways. And we want to do this in human cells. And then using this system, we want to identify FDA-approved drugs that can restore the disrupted pathways. And we're extremely grateful to the Brain Foundation for funding this idea that we have. So autism is a neurodevelopmental disorder, which means it affects the developing brain, one of the most complex organs in the body. The part of the complexity of the brain is its many cell types, some of which are shown here, and all of them thought to play a role in autism. So for each of the ASD risk genes, we really need to understand the function of these genes in each of these brain cell types. Unfortunately, the human brain is inaccessible to us to study these cells directly, but a workaround for this is something called reprogramming that won the Nobel Prize a few years ago, where you can collect blood or skin from individuals and then revert them back to what we call induced pluripotent stem cells. And then in a process called differentiation, we can direct these cells to become any cell type we want, including cell types relevant to ASD. So this is the best system to study human neurons, especially for neurodevelopmental disorders. So the landmark ASC paper that Dr. Buxbaum presented found that many ASD risk genes are involved in gene expression regulation and neuronal communication. And what you can see here is if we look for their expression over the course of brain development in humans, you see that many of them are strongly expressed in the developing brain, which is nice because we can use the induced pluripotent stem cells that I described to generate these cell types in a dish. And we've done that using the iPS cells. So here I'm showing the neural progenitor cells that we've differentiated from the iPS. This is what they look like by bright field. And then we can stain them for various markers of neural progenitor cell identity, which is shown here. So we've generated these, and this will form the basis for our assays to study the function of ASD risk genes because it matches their developmental expression. But the top goal of the project is to study many ASD genes at once so we can compare their effects and find convergent mechanisms. So on this slide, I'll tell you about our plan to study these genes at once. So the foundation of the project is a technology based on CRISPR interference. And so here's an example of an ASD risk gene being transcribed in its into its mRNA. So we can use this CRISPR interference technology to direct a protein called dead Cas9 to the start of this gene and thereby block its expression. This mimics a loss of function mutation, which many of the genes that Dr. Buxbaum talked about act through loss of function mechanism. So the critical piece of the CRISPR-Cas9 is a short piece of RNA called a guide RNA that enables us to direct this Cas9 anywhere in the genome. And importantly, this makes it really high throughput because we can generate vast numbers of these guide RNAs targeting each of these individual genes. So now I've described our IPS system and our CRISPR-I technology, and now we're going to put it together to make a platform to study many ASD genes at once using this. So we've generated a guide RNA library targeting 75 of the high confidence ASD risk genes. Another feature of this technology is that when you add it to the cells, we can add it to neural progenitor cells so that each cell gets only one perturbation or one of these ASD risk gene targeted, as shown here by kind of independent colors in each cell. And then we can differentiate these neural progenitor cells into neurons or different types of neurons or any cell type that we think is relevant to ASD, like OPCs, for example, although that's beyond the scope of what we've proposed so far. So now we'll have a pool of these cells, each investigating one ASD risk gene, but studying many of these genes at the same time. So how do we tease out the effects of each of these things? We use another technology called single-cell RNA sequencing, in which we put these individual cells into these tiny droplets and then perform RNA sequencing on each of these separately. So single-cell RNA sequencing allows us to generate the transcriptional signatures of ASD risk genes. So gene expression profiles, they represent a high-dimensional signature of cell activity, health, and identity. So we can use these signatures. So this is an example of a signature for each of the ASD risk genes that we've obtained from single-cell RNA sequencing. So we can use these signatures to see which biological pathways are commonly disrupted by ASD risk genes. And these signatures are going to be in a cell type specific and time point specific manner. So importantly, and the whole reason we're doing this method is because we can now compare many of these genes at once to find patterns shared between these genes. And these patterns represent convergent mechanisms across these genes. So instead of targeting each gene separately, we can now think about targeting groups of genes that act the same way. So I'll get back to that in a second. But I first wanted to show some preliminary data that this method works. So in work that I published last year, I demonstrated this approach looking at 14 ASD risk genes, and I found that 5 of the 14 that I studied, as shown here on the left, had strong overlap of their transcriptional signature, which means they shared downstream target genes. When we investigated which pathways were affected by these genes, we found some gene ontology terms that suggested that neural progenitor cell differentiation and proliferation were affected, which is again why we're using the human iPS neural progenitor cells in the differentiated neurons now to follow up these findings across 75 of the top ASD genes. So going back to our single cell transcriptomes and thinking about convergent targets for therapeutic intervention, one of the emerging ideas for drug discovery is reversing gene signatures. So here's a schematic of that, where you can see here that genes B and D have the same signature. So now what we can do is look for drugs that have the opposite signature in the same cells. And the idea is that these kind of cancel out, so that drug X, by adding it to cells with these mutations, would actually reverse the signature and push these cells back to a healthy state. And we can do this now because all of the FDA approved drugs have now been tested in iPS neurons and generated signatures for each drug. So all we need to do, using some elegant algorithms developed at Mount Sinai, is to look for these drugs that can reverse the signatures. So this is one of the main goals of our project, and hopefully it will lead to some therapeutic discovery. But we can also ask some other questions in our single cell data. Without getting into too much detail, and for more details, you can refer back to my 2020 paper, but we can also use this data to investigate whether ASD risk genes alter the pace of neuronal differentiation. So our data ends up looking something like this when you plot it in this pseudotime analysis, where you can see that differentiating cells choose their fate. So because we can see these fate choices, we can then ask, do ASD risk genes contribute to alterations in cell fate? So one of the leading theories in autism is a disruption of the balance between excitation and inhibition in neuronal circuitry. And this could simply be from generating too many or too few excitatory or inhibitory neurons. So we would be able to see that in our single cell data. So, and many of the ASD risk genes might act in the same way or in opposite ways, allowing us to come up with separate therapeutic targets for each of these sets of genes. So that's kind of the big picture view of everything that we're working on, or not everything that we're working on, but what we proposed in this Brain Foundation grant. So I'll end there with a view from our lab. And I want to acknowledge the generous support of the Brain Foundation. I'm also supported by the Beatrice and Samuel Seaver Foundation, BBRF, and I also want to thank the ASC, the NIMH, and my team and colleagues. And I'll let Dr. Buxbaum take over again, and I'm happy to answer any questions in the chat. Thank you.
Thank you, Matt. Yes, we're happy to answer questions.
Thank you, Joseph. Thank you, Matt. There are a couple of questions in the chat. It looks like one for each of you. So Joseph, we'll start with the first one, and I'll just read the question. If an environmental factor is driving the rise in numbers of autism, so I guess maybe you can just address that premise before I go on with the question. So if the environment is driving the increase in the numbers of autism, wouldn't it make sense that as time goes on, it would present as if a genetic subpopulation, which is simply reacting to that factor, is more likely to have autism, whereas the true causative agent is the environment? How do genetic studies circumvent this problem?
Yeah, so that's a good question, and there are a bunch of good answers. But first, I think most people in the field would say that the rate of autism increasing is largely at least 30-fold increase simply by changing diagnostic criteria. But genetics actually can handle an environment, and we actually formally model it. There's a famous quote by a British epidemiologist and geneticist who said, if everybody smoked, lung cancer would be a genetic disorder, right? So if there is a consistent environment that, everybody shares, there's no change in the environment, you only see the genetic effect. If there's no change in the genetics, you only see the environment effect, right? So height is one of the most genetic traits we have, and yet people over the past 200 years, everybody gets taller and taller, right? It's because there is very little change in the environment over the course of a study, but there's big changes in environment affecting height over the course of decades and centuries. So we're actually able to do a pretty good job of teasing apart environment versus genetics. And but I would say the most important thing is that those exact things, whether it's people say it's 60% genetic and 40% environment, those are pretty meaningless because depending exactly on what's changing, it could become very different. The important thing is whether there's any genetic component, and if so, can you find the genes, or if there's any environmental component, can you identify them? And that's really the important thing. And whether, some people say it's 50% genetic or some people say it's 80%, in biology, those are the same number. biology, biologists have what's called physics envy. We're happy if we get the sign right. We don't have anything down to 4 decimal places yet.
Oh, a plug for quantitative biology there at the end. I think this next question is for Matt, so thank you so much. What's your opinion on bone marrow stem cell infusion via cerebral spinal fluid?
I think there's been a couple of studies that looked promising. I think the largest one failed to replicate. I think if they're safe, people should. I think they all need to be tested extensively for safety before we can even talk about anything with efficacy for these, just like any drug. But hopefully there's some therapies emerging on the near-term horizon, wherever they may come from. I don't have a specific opinion on that, though.
I will just add that the stem cell work that we talk about is different from stem cell therapeutics, right? In stem cell therapeutics, you're putting in cells into somebody under different circumstances. Here, we're using stem cells to make brain cells in vitro, so we can study brain tissue without actually doing a brain biopsy. So the stem cells here are used because stem cells are able to make other cells, any other cell in the body, ideally, and so you can create human brain cells in vitro in a petri dish, and that's how we use stem cells, which is very different than the clinical application. But as Matt said, there's not been anything very compellingly positive. Most of it negative, and one of the ones that generated some interest a few years ago was just retracted because of significant ethical concerns as well. So I would say we don't have actually any affirmative evidence for stem cell therapeutics in autism.
And one final question, I think this is also for Matt. Can this be tried on individual patient sample? I'm assuming it's the iPSCs, especially, say, very positive responders from previous trials to validate?
Yeah, that's a nice question. And it's something we're doing in parallel, as Dr. Buxbaum showed in the table. We have several teams, but it takes teams of individuals to study just one of these disorders from one set of patient-derived IPS lines. Whereas my approach, a single person could generate this whole library and study all of the genes at once. So in terms of learning the function of the genes, I would recommend my approach. But yeah, then validating any findings, it would be valuable to have the patient-derived lines as well. And pharma is especially interested in the actual patient-derived samples for their validation. So I think they're nicely complementing systems.
Terrific. Thank you, Matt. I have a couple of questions as well, but in the interest of time, maybe we'll move forward and I'll track you down offline. Thank you, Joseph. Thank you, Matt. So now we'll move to the talk by Stuart Lipkin. Stuart is the Hannah and Eugene Stepp Chair and Professor, as well as co-director of the Neuroscience Translational Center in the Department of Molecular Medicine at the Scripps Research Institute. And the title of Stuart's talk is Potential MEF2 Transcriptional Activator to Improve AST Phenotypes.
MEF2C Association in Europe, many of them are on this teleconference due to the magic of Zoom. So you're truly international today. So I'm going to talk a little bit about, well, first, thanks for having me back. It's always nice to be invited back. Maybe that means that you did something good the first time. So I will catch you up on what I said last year, and then we spend most of the time on this MEF2 transcriptional activator. And I just want to give a shout out to Matt, because I was able to remove half my slides because he gave a lot of the IPS and CRISPR-Cas9 that I was going to talk about. So that's fantastic. I can just show you the results without having to worry about that. Matt actually mentioned this, that excitatory inhibitory imbalance is thought to underlie many of the abnormal phenotypes in ASD. I'm having an issue with my screen here, but I think I fixed it. The transcription factor MEF2C is involved in multiple forms of autism and intellectual disability. And so we hypothesized that boosting its activity might normalize EI imbalance and neurobehavioral abnormalities. And we screened what's called the Scripps Reframe library. The Scripps Reframe library is a unique library that Scripps chemists, Scripps, I am so honored. Scripps has, it's been ranked 1 or 2 in the world in chemistry. The chemists think I know some chemistry, so we're all going to be quiet about that. And they put together a library of not only all the FDA-approved drugs, as Matt had mentioned, but all the EMA-approved drugs in Europe and other compounds all over the world that have made it through phase 1 safety trials, even if they're not yet on the market. So the beauty of that approach is that we have all the drugs basically that have been ever tested in human that are safe, and we are able to test those. And the lead candidate I'll show you today has extensive experience in humans for another indication and shows excellent MEF2 activation and corrects EI imbalance. And I'll show you some of that data. Okay, so this is bringing coals to Newcastle. You've heard this in multiple other talks. The latest CDC statistics are now 1 in 44 children. You've also heard Dan Geschwin's name in both Sarkis's talk and Joe's talk. There are multiple slides. Dan is also my colleague, and he looked at, I'll show you the data in a moment, a series of ASD genes and was able to parse those genes into families. And interestingly, the transcription factor, MEF2C and MEF2A, a related transcription factor, control many of those genes. Dan then contacted me. I was the discoverer of MEF2C, and with Dan, we've been looking at MEF2C in various neurologic diseases, including autism spectrum disorder with intellectual disability. So our reasoning is that therapies aimed at improving MEF2C-associated ASD phenotypes might potentially affect many other forms of ASD, because we know, not from iPSC data, although we do know, from real human data, that MEF2 controls many of the genes involved in ASD and intellectual disability. So here's some of Dan's data from a paper in Cell, and you can see these 5 hubs of genes that are many of the genes that Joseph showed you in the prior talk. And MEF2C and MEF2A are 2 of a few transcription factors. Transcription factors are proteins that go in and turn on genes. So they control many of the other known autism genes. Now, interestingly, MEF2C itself can be mutated and form a very severe form of autism and intellectual disability. We first discovered this actually in a mouse, and then colleagues subsequently discovered it in humans. And so this is a gene that encompasses the MEF2C region, and now we know specifically of many mutations in MEF2C that cause this. If you look at the UK database and some other databases, it has the possibility of being extremely frequent, in that more frequent than, say, Rett syndrome. Oops. And these children generally have severe autistic behavior. Only a few of them talk, accompanied by epilepsy, periodic tremor, and abnormal EEG. We'll get back to the electrical activity in a minute, and often severe intellectual disability. Although there are a few that are less severely affected that I've seen. I'm also a physician and have seen, it must be nearly 100 of these cases now, and I'm in contact with many of the families. So we have 2 disease models. One you just heard about. We also use human patient skin biopsies, both from patients that we then use CRISPR-Cas9. We take tractable mutations. These are mutations that are not too large that we then can correct. We've also introduced mutations into the normal genome to mimic MEF2C. So we have a whole family of these MEF2C mutants. Importantly, we've used multiple genetic backgrounds. You must do this to ensure that it's not something in the background that's causing your phenotype. So we have them from multiple families that we've corrected. We also have a MEF2C mouse that we published, shown here, and we use that to test our drugs. Although, like Matt Lally, we feel it's really important to do this in a human context in iPS cells. Okay, so let me show you our disease in a dish model of the human MEF2C haploinsufficiency syndrome. And so, as Matt just eloquently explained, so I was able to take out many of my slides. We take patients, make human iPSCs, then we can differentiate them into neuroprogenitor cells, into neurons, and multiple other cell types. We've done that not only in two-dimensional cultures, but in these 3D mini brains called cerebral organoids. I'll show you some pictures in a moment. And this just takes you through the stages. These show that we've made NPCs, that is pluripotent cells. We then can take them through the various stages to make neurons. They start to look like neurons, but importantly, they stain to show their neurons. They have the electrophysiological characteristics of neurons. We also have done single cell RNA-seq, and I'll tell you about that in a minute. So I think the most important finding from this, which should be appearing shortly in Science Translational Medicine, is that the MEF2C ASDs make a lot more astrocytic cells and fewer neurons. So here MAP2 is a marker for neurons, and you can see there's not a lot of green here in the MEF2C child's neurons. In wild type, estrogenic wild type, a lot of neurons. And the opposite is true if you look here. GFAP and S100 beta in this context are astrocytes, and you can see more in the MEF2C neurons after 6 or 10 weeks in culture, and fewer in the wild type. So you're not making neurons properly, also not making synapses properly. Yeah, so I mentioned we can make these 3D cerebral organoids in a dish. We've actually brought some of these kids in, told them we created a little piece of their brain or part of their brain in a dish. They look like this. Many of them actually have little ventricles, what you have in your brain, as you can see here. They have layering. I won't go through all the different layering we have, but we get them pretty mature. We've kept them for 6 or 9 months. We do this in multiple diseases, Alzheimer's disease, which I'll come back to at the end, actually. But I'll show you the autism studies today. And then single cell RNA-seq shows multiple cell types, interesting deficits in both excitatory and inhibitory neurons in the MEF2C ASD patient samples versus wild type. And others have shown that MEF2C is very important in both excitatory neurons and inhibitory neurons. Interesting also in human microglia, although I won't talk about that today. But we think that underlies some of the infections these kids get. Okay, so let's cut to the quick. What do these cerebral organoids do? Well, they manifest EI imbalance, excitatory inhibitory imbalance. They actually have too much excitation. And this is one indication. We have patch clamp recordings. This is calcium imaging. We have multi-electrode arrays where we can record the whole brain like an EEG. I just want to show you calcium imaging. I showed some of this last year, but we have a lot more to show you now. And so you can see this is the normal wild type, and the neurons are signaling to one another. And we can quantify this. I'll show you the quantification in a minute. If you look at a kid, a child, iPS cells, this child happens to have seizures, and whoops, we've actually recreated the seizure in a dish. You can see it pulsating like that. We can quantify that. Not all of them have seizures. Sometimes they just have too much electrical activity. So the EI is balanced in this case toward E. Too much excitation, you can see it here. Last year, I talked about a drug that we're developing called nitrosynapsin, and you can see when we put it on, it calmed things down dramatically. Here it is again, we calm it down, see everything gets much more toward normal. So as I mentioned last year, this is an interesting type of glutamate receptor antagonist called the NMDA receptor. It's based on a drug that I patented actually for Alzheimer's disease. It's now one of the FDA-approved drugs for Alzheimer's disease called memantine. We modified the structure of memantine, and it has a second site of action at a site we call the S-nitrosylation site, more chemistry that had been discovered in my lab some years ago. So this is a dual-acting, very, very potent drug, but very, very safe. Little company in Boston is developing it. Here's an update on that, and then I'll get to the MEF2 activators. So I won't show you again, but I showed you last year, and we published it, that this drug, nitrosynapsin, damps down only the excessive excitation. It doesn't affect normal excitation in the brain. It also corrects multiple neurobehavior abnormalities in our MEF2C mice. We've now nearly completed pre-IND studies in preparation for a human clinical trial. We're actively seeking financial support. There's a major philanthropist who's involved in autism who's considering funding the trial, but we'd love to include any of you if you're interested. We are actively raising funds for the trial. We're going to do a phase 1/2 trial at the same time. And we've identified the first site, which will be at Mass General. They've developed a clinical biomarker that reflects this excessive NMDA receptor activity, so we can get an early readout with our biomarker in addition to doing neurobehavioral tests. So that's the plan for nitrosynapsin. Let me now turn to this MEF2C approach, which we're very excited about, and which the Brain Foundation and the MEF2C Association have been funding recently in the lab. And so I mentioned that obviously MEF2C haploinsufficiency, where one copy of the gene is missing or has a deletion or is somehow not working, there's one allele, that the other copy of MEF2C that's still there. Perhaps we could overstimulate that and compensate. And then most importantly, because Dan Geschman had shown that MEF2A and C are very important in controlling those hubs of many, many other genes involved in ASD, we reasoned if we could increase MEF2 activity in general, we might be able to turn on those genes and offer some benefit. And so we carried out high throughput screening of the chemical libraries I mentioned at Scripps, not only that Reframe library, but multiple other libraries. We have about 1.5 million compounds. I haven't screened all of them. I don't believe in that mindless screening. We screen for drugs we think are in categories that can work. So we have about 100K screen that we've done. And we've identified 5 drugs that are existing drugs, already been in humans. And we have one candidate, I'm going to call it compound Y today, and I'll show you some data with that compound. So how do we do this? Well, as I mentioned, MEF2C and A, and there's other forms of MEF2, but those are the 2 that we know are involved here. They bind to a piece of DNA and turn on various target genes. What we've done is replace one of those target genes with luciferase reporter, something that bioluminescently has bioluminescence, so we can see it in the screening method. And then we add multiple drugs, and we see if it activates MEF2. And here it's shown in a 384 well plate. We have a luminometer to read that bioluminescence. We can put one drug in each, and we get basically certain drugs turn on MEF2 activity, shown here on the y-axis. We have what's called a very good Z-score. We have a good, anything over 0.5 means you can go and screen your libraries, which we've done. So here's one typical screen of many that we've performed. And this is MEF2 activity. Remember, turning on that luciferase. So we're going to turn on all those genes that are turned off, not only in MEF2C haploinsufficiency, but in many other forms of autism. And you can see the top hit here is this compound Y. We do have other hits, but we're particularly interested in this one because it has very extensive use in people for another indication. And so now we more precisely look at MEF2C activity for the aficionados out there. We we compare it to Ranilla. So we just want to make sure it's not turning all transcription on, but just MEF2C. Ranilla is to monitor background activity, if you will. And we have very good activity at MEF2 sites. So we would turn on MEF2A and C in point of fact. Now we wanted an indication. Remember I said the neurons weren't growing properly and they weren't growing out their neurites and their synapses. So we have multiple ways. I'm just going to show you one of them. We can actually look at that neuronal marker MAP2, which is all over the neurites, and we can quantify that as shown on this plot here. And what you see is there's more MAP2 when we treat the haploinsufficient neuroprogenitor cells that have less MEF2C. And you can see much more MAP2 protein. I won't show you pictures, but also we get many more neurites and synapses when we put the MEF2 activator back into the dish or into a cerebral organoid. Okay, so remember I showed you this picture of a child with seizures, boom, boom, boom. We can actually quantify that aberrant electrical activity, giving you EI imbalance. And that's kind of shown here. So wild type, you do have normal electrical activity. Here we're just looking at those calcium bursts. And you can see in the MEF2 patients, this is patient 1, but as I said, we have half a dozen patients, you get these bursts of calcium activity. Whoops. And when we put on compound Y, we still get the normal activity, no more bursts, and it's quantified here. So we are able to rectify EI imbalance. We're now testing this compound to look at various behavioral phenotypes. So we're really excited about it because it's been in patients. It's interesting, it's not FDA approved. So that also gives us an intellectual property approach to it, so we can get some company interested in bringing it forward. So in summary, what I told you about today, and I was able to go quickly because Matt, fantastic job, Matt, in kind of giving a lot of the background. I could skip those slides. So we were looking at MEF2 haploinsufficiency syndrome, and particularly we're interested in this excitatory and inhibitory imbalance as a biomarker, if you will, of drugs that might improve not only MEF2C haploinsufficiency syndrome, but other forms of autism that have EI imbalance. Now, just amazingly, my close colleague at MIT, Li Wei Tsai, published in Science Translational Medicine last month that MEF2C is also important in various forms of dementia. We had been worried if we played with MEF2C later on after initial development, we actually might do damage rather than help. And I think that's a real worry about genetically interacting with any of these genes that Joe or Matt or I have been talking about. But here we have Li Wei, who's vetted this for us, and she's actually found in the oldest old people who have a resilient Alzheimer's or other forms of dementia, they have more MEF2 activity. And in a mouse model, if you decrease MEF2 activity, you get a form of dementia. Multiple mouse models of dementia have left MEF2C activity. And if you genetically increase MEF2C activity, you can abate the dementia phenotypes, memory, cognition, and multiple neurobehavioral tests that Li Wei and her group did. So we're now testing our drug in Li Wei's dementia models as well. And so what does this mean? Well, we think this EI imbalance, and we recently published a review on this in Annals of Annual Review of Pharmacology, is that at the extremes of life, life is a bell curve. We're all somewhere on that bell curve. At the extremes of life, your nervous system is very, very susceptible to EI imbalance. And it's not just the neurons. As other speakers have mentioned, other cell types, both the myelin-forming cells, the astrocytes, microglia, they're all involved in this EI imbalance in an intricate network. We know that, so is the extracellular matrix. And we think you're vulnerable, and you paradoxically, at the extremes of life, can develop this EI imbalance. And some of the drugs we use in one disease, say in Alzheimer's, might be amenable to being used in autism to correct the biological pathway that's being disrupted. And I showed you that the MEF2 activator compound Y increases not only MEF2C transcriptional activity, but this biomarker phenotype of EI imbalance. This is a drug that's been in people, has no significant toxicity, and so we're really excited about bringing it forward. So I've had tremendous help. Here are the people that have helped me, other labs as well. Here's Dan Geschwin, Jeff Rosenfeld here at UCSD, Nick Short, my bioinformaticist. I particularly want to acknowledge and thank Pramila at the Brain Foundation, who had the faith in us doing this at a very early stage of the work. Thank you. And Sarkis and Joe, whoever votes for this, thank you all very, very much. And also I want to acknowledge the MEF2C Association in Europe that I mentioned, which is a fantastic group of families with a tough disease, with MEF2C haploinsufficiency, who've banded together really all over the world to try to help with this kind of work. So this is where the work is done. It's a beautiful building right opposite the ocean. I better not talk about this. I know the weather back east isn't good right now. And you're all welcome to visit after COVID, please. And I'm glad to take any questions. Thank you.
Thank you, Stuart. That was really enlightening. And I would encourage people to ask their questions in the Q&A. There's one question, maybe I'll start, and Matt, feel free to jump in as well, but it's more of a general question on the iPSCs and how well they can model the diversity of neurons found in the brain, human, animal, what have you. I guess that's the first part of it. And secondly, to what degree does this correlation or lack of correlation with the diverse neurons, whether it's at the expression level, the activity level, the neurotransmitter level, to what degree does an intervention like a drug or a CRISPR intervention inform us about what neuronal subpopulations may be relevant in the human brain?
Yeah, so let me start with that, and then we can get Matt to chime in too, if you don't mind. That'd be really good. Yeah, you're right. Look, we have a developmental model in a dish. Luckily, this is a developmental disease, so it's not that we're trying to model a very old disease. Now, my colleague, Arnie Kriegstein, who has worked on development in iPS cells for a number of years, has wrote a series of papers and also editorials, if you will, about this question. Can you model in a dish a human disease? I mean, you're in a dish. So there's several issues here. One is the cells don't develop totally normally, even in the brain organoid model. They develop more slowly. And, I think particularly this is important for neurodegenerative diseases. You're 65 or 70 years old, not like autism when you get a disease. But interestingly, in the dish, they're under a certain amount of stress. And that seems to speed up the development. So even Arnold has agreed with us that we can mimic many of the features of the disease. But this is why having a biomarker of the disease that mimics the phenotype, like EI imbalance, is so critical here. What I'm telling you is I can, in a dish, mimic a finding that I found in mice and others have found in EEG in patients. And I can use that biomarker to use my drug discovery. So like Matt, we're also doing something called CMAPS Link 1000, where we have thousands of drugs that fix transcriptional activities, and we can screen drugs that way. I think that's fine. Matt, I actually didn't know that it had also been done in human neural progenitor cells. I may write to you to get some more data on that, because the database I have is not just for human neural progenitor cells. Now, let me address the single cell RNA-seq data. We do see a diverse number of different cells in our cerebral organoids. Okay, now I can't tell you we have every type of neuron, but we have looked against adult databases, and that's actually how we classify the cells. We look at a transcriptome signature, if you will, of, say, an excitatory neuron in the striatum. And we can match those, and we do see a vast repertoire of cells, but I know we don't see all the cells in the cerebral organoid, but we do see many of them. And so we're encouraged that because we have that phenotype, that electrical phenotype that we're very good at monitoring, we think we have some concordance, if you will, on the disease.
Sure, thanks. A couple more quick questions, I think. Is a planned trial for compound Y in patients with MEF2C mutations or in a different subset of ASD?
Yeah, so that's great. There is no, although we're working with colleagues to get this, we really don't have what they have for Rett syndrome, which is a natural history study of MEF2C, which would really facilitate our doing a clinical trial. While we'd like to do that, I think all of these drugs, and we've met with the FDA and discussed this ad nauseum, we're probably going to be doing it in another subset of ASD patients that are a little more tractable. So both for nitrosynapsin and the MEF2 activator drugs. That's the plan right now, simply because the FDA is driving us that way, but we hope in the future we absolutely want to treat those children. Look, all of our children need help with this, and we're working hard on that, as we all are at the symposium.
Yeah, I could imagine having a pre-screen based on a narrow genetic subpopulation, which is make recruitment so challenging that it would be very difficult to even run the trial, right? I'm just wondering, maybe just dovetailing off that question, because MEF2C controls so many other genes, are there other either genetic or maybe physiological parameters one can use to enrich for a population that may respond to the drug?
There is, as I mentioned, the Mass General group has developed a biomarker that they'll be publishing and talking about soon, and it actually reflects the phenotype I showed you. And so we're very, very excited about that biomarker, because when you have a little company taking a drug forward, and I've done this several times now with memantine and other drugs, you have to kill a drug. You have to, this is the trick of a little company. You don't make a drug, you kill a drug, so you get to the right drug eventually. So having a biomarker for an early readout in a study is absolutely critical if it's a reliable biomarker. And we think we have one.
Joseph, you have a question?
Yeah, I was just wondering about, some of at least the nitrosynapsin and even this new compound Y, they seem to be working on, as you said, excess electrical activity. What about trying it in just epilepsies? Intractable epilepsies, for example.
Yeah, well, that will be one of the readouts. Many of these kids have epilepsy, so that is one of the endpoints we've discussed with the FDA.
Thank you.
Great talk. One more question in the Q&A. Does nitrosynapsin activate Mtor?
Well, downstream it may. What it does is, I haven't looked at that specifically, but we know that it, we partly have looked at that. So we've tested it in a tuberous sclerosis model, and it's published. And I'm not going to be able to conjure up that immunoblot in my head right now. We've looked at the pathway and it corrects the pathway. It does interact with the mTOR pathway. So the answer is yes. And we think that's because it's in part, many of these genes are neuronal activated genes. And we know that MEF2 damps down this excessive electrical activity. Interestingly, it spares normal synaptic activity. That's why it and memantine that I developed previously are so well tolerated as opposed to many other drugs that have tried to interact with the NMDA receptor.
Sure. And Joseph, is that a legacy hand that's raised, or do you have another question?
That's great. Sorry, I don't remember that one.
We'll move on. Thank you so much, Stuart.
Thank you all. Thanks for having me.
Absolutely. To the next talk by Kazu Takahashi. She's an assistant professor at the Department of Radiology at the Gordon Center for Medical.
Okay, can you hear me? Hello?
Yes, we can.
Oh,
Yes.
okay. Okay, so, first of all, thank you so much for having me and bring me here to give to share my story about manus binding lectin MBL. So I wasn't sure what to do, but so I decided to give kind of broad sort of background what MBL and my thinking about MBL at this stage. So at this point, MBL to me, it's multifunctional. I hope I can convince you what I'm telling you is true. And MBL is like sentinel, so providing constant surveillance through, and also kind of house cleaning, and also through coagulation, I would like to call it bionic glue. And lastly, it is a disease modifier. So as long as you don't have anything, you may not feel it, or you wouldn't know till you look for it. But if you have underlying problem or some infection, then MBL deficiency in particular would make you feel something wrong. Now, I'd like to start with a big picture, at least to me. So thanks to host defense system, we have innate immunity, which kicks in second or minutes really after our body is exposed to danger, could be just injury or invading pathogen, and provide local containment. And one of this system is called the pattern recognition molecule, which MBL belongs to. And if we now this sorry, oops, sorry, so oops, sorry, I'm messing about. Okay. Now, innate immunity has linked to acquired immunity, now which takes at least a week, a long time, but provide systemic and target-specific protection, often quite for lifetime, since it has memory component in it. Now, what is MBL? It belongs to the collectin family, and as I mentioned, pattern recognition molecule of innate immunity. By itself, functions as opsonin, which facilitates phagocytosis by so-called professional phagocytes like a neutrophil and macrophage. And also in cells, has served as a post-translational glycosylation, so located at border of ER and Golgi, which was actually done by the other research group. And also MBL in complexes with a mass, MBL associated stem proteases activate the lectin complement pathway and also function like thrombin. Now, I mentioned innate immunity target is broad. So when we started MBL target, we really thought only pathogen. So bacteria, fungus, and virus. But we now know MBL can recognize much broader sugars, and ourselves, like once it's injured or transformed like a cancer, Mbl can recognize. Also some enzymes and amyloid beta, like a Alzheimer protein. Now, as a function, by itself can serve as opsonin, so increase phagocytosis by neutrophils and macrophages of Mbl target. And in a complex with MASP1, MASP3, so Mbl functional Mbl is hexamer. So then this complex activates the lectin pathway. So halfway through alternate pathway as well as classical pathway. Now, our contribution to this was this MASP1 Mbl complex can function like thrombin, meaning transform from fibrinogen, soluble form to fibrin clot. And the Mbl is primarily synthesized in the liver, but some small portion expressed in small intestinal mucosal epithelial cells. And I personally believe it should influence on the microbiome. We heard earlier this morning as well as yesterday. And even though Mbl is synthesized in the liver, most circulates in the blood and seeps into tissues. So far, Mbl has been identified in CSF and alveolar lavage and even in the skin. Now, I'd like to talk about the molecular structure of Mbl. So starting from 5', so-called system-rich domain, which we think, well, actually it is, to bundle monomer of Mbl, and followed by collagen, long collagen region. Hence, it is called the collecting. And CRD, carbohydrate recognition domain, which is primary target recognition domain. And Mbl has 4 exons, and of which first exon, towards 3' of first exon, the neuron SNP, they're called D, B, or C allele. So if you have any of these, Mbl secretion is reduced. And in addition to this, at least 3, actually we know more, 5' open reading region as well as the 3'. And because of these alleles, we have so many haplotypes. This ended up in broad range of plasma Mbl levels from undetectable to, we've seen 20 microgram per ml, which is a lot. But average, if you are wild type for both alleles, 2 microgram per ml. And if you have one heterozygous or homozygous for those amino acid substituting mutation, less than 1 microgram per ml. Now, this is the EM. We never published, but just so that, I think you can have ideas. So that we usually call it like a bunch of tulips. It's a hexamer, and probably this one's much easier. You can appreciate the 3 of them. So when we started Mbl, I've been working on this couple of decades, and only interest was infection. So the first report was the children who have recurrent infection had low or undetectable Mbl. And soon afterwards, the life expectancy of cystic fibrosis patients was shorter in Mbl deficient people. On the other hand, these Mbl deficient people are better against intracellular pathogen. At the time, we knew only 12 TB. So because of this clinical notion, we generated Mbl knockout mice. Now I have to mention gene compared to mouse and humans. So mouse, they have 2 protein derived from totally different chromosome, Mbl A and Mbl C. MBL-A is acute phase protein. So, and while MBL-C is consistently expressed. So when it's acute phase protein, so the naive mice, MBL-A is not really much, but once like LPS challenge, then MBL-A shoots up. Now compared to this, human, we have only one protein, which is genetically homologous to MBL-C. So often some people might describe MBL-C, human MBL-C. But, and if we compare the protein level, actually 40–50% homology between these three human, mouse, A, and C. So when I'll give you phenotypes, I'll talk about Mbl knockout phenotypes. So Mbl knockout mice are actually double knockout of MBL-A and MBL-C. So we made single knockout and cross to make double knockout. So MBL no mice. So I'd like to mention, so there are sounds very similar molecules out there now. First of it, it's LMAN1, which is also called ERGIC53 or Mannose receptor, which we also worked on. I'm not going to talk anything about this, but both these sound cruelly similar molecules are on membrane protein. And so when we made Mbl no mice, mice were healthy and fertile. So this was really great for us. And also we didn't see any obvious phenotype even at the histological level. And we made sure there was no transcript, no protein, and lectin complement pathway was undetectable. Most of the talk I'm going to share are already expressed. So if you would like to see detail, please read the original. Okay, and so first of phenotypes we expected, yes, Mbl no mice are susceptible to the infection and impaired acute inflammatory responses. So I would like to share two cases. One, Staph aureus blood-borne infection. So we inoculated mice with Staph intravenously, and red column for MBL null and gray for wild type. So this data is bacterial titer 2 hours post inoculation. So you can see significant, at least 1 magnitude more bacterial titer in MBL knockout mice compared to wild type mice. And at this 2 hours, we also looked at cytokine TNF alpha IL-6. Both were diminished in MBL no mice. So, and second infection example was a viral infection, influenza A virus infection, and is the primary lung infection. And in this data, MBL no mice are in closed bar and wild type in open bars. So day 1 post infection, still MBL knockout mice lungs could not clear a virus, while wild type almost kind of undetectable. And we also looked at, just part of inflammatory response, we looked at white blood cells and percent of neutrophils. You can see response, these white cell numbers and neutrophil populations in MBL lungs were different from wild type. And I was drawn to imaging all the time. And at this time, I had only FDG accessible. But I did chronological imaging post-infection day 1, day 4, and day 8. Left panel showing wild type mice and the right panel showing MBL null mice. So by day 8, there was almost no signal in the wild type mice lungs, but you can see some activity, FDG uptake activity in MBL null mice. So supporting again, Mbl neural mice have aberrant response to inflammatory response. Now, based on the Mbl pathogen, broad pathogen recognition, there is a blood cleansing. It's like a dialysis, but the Mbl cartridge is incorporated. So the idea is to clean pathogen from blood stream. It's early stage of clinical trial. And now from here, I'd like to share with you the phenotype we didn't expect, because we didn't really expect much actually. But surprisingly, first of all, Mbl neural mice responded greatly to polysaccharide vaccine. And so we had a collaborator at the time working on glucose strep type 3 polysaccharide, which was conjugated tetanus toxoid. And altogether, we immunized 3 times, 20 days apart. And this IgG titers are measured at day 60. And this data shows you glucose strep polysaccharide 3 specific IgG. We also measured just the whole total IgG, IgM. We didn't see a difference between wild type and Mbl. So I'd like you to focus on between wild type and Mbl, and increased IgG isotype was IgG1. And then this increase was in part increased total peritoneal cell number. So peritoneum, that's where we immunized. We used to immunize, and B1 cell was significantly increased in Mbl normal mice. And so the next phenotype was quakeropathy. So Mbl neural mice had a prolonged bleeding time. And now in mice, you can do kind of simple experiment. So you cut 2 millimeter tip of a tail and put in 37 warm PBS and time till you stop seeing the blood is coming into PBS. So most wild type stops less than 1 minute. While Mbl neural mice, many of them didn't stop even 15 minutes. And at this time, we wanted to make sure, the other coagulation factors are not altered in these mice. And as far as we looked at, like platelets numbers and platelet functions were comparable to wild type. And more so when we pretreated these Mbl neural mice with recombinant human Mbl, the phenotype went back. So we could say this prolonged bleeding time was due to lack of Mbl. And C3, now by the time we had a mice, people start saying, oh, Mbl is a complement protein, but this result shows you this prolonged bleeding time was C3 independent. Now, as I mentioned earlier, Mbl forms a complex with MASP1. And so we subjected to the same experiment, MASP1 knockout, MASP1 null and MASP2 null. I'm saying MASP1 null, but actually, so what MASP1 null is MASP1 3 double null, and MASP2 null is MASP2 and SMAP null. So as you can see, MASP1 null mice had prolonged bleeding time, but not MASP2. So we conclude that this prolonged bleeding time quakeropathy was due to lack of Mbl and or MASP1. And this work was done by Peter Gall's group. So MASP is directly linked to pro-inflammatory pathway. So here is the Mbl target and Mbl MASP complex, and PAR, protease activated receptor. There are 4 of them nowadays. And so what happened is when this complex binds to the target, Mbl target, MASP can activate PAR and seems to like PAR2. By the way, PAR1 was also known as thrombin receptor. And this PAR activation actually directly linked to pro-inflammatory cytokine signaling pathway. So this is a link between MBL being acute phase and at the molecular level. And the next was bound phenotype. So we used dry bound, so we burned the back of mice. And in the wild type, what happened is ESCA or the burnt skin slough off overnight. But it was not, oh, this is a picture. So it's kind of expensive, but I was told that mice don't really feel pain. Well, any case, so the wild type ESCA shed off, so you can see muscle, back muscle, while MBL no mice didn't. And so most 21 out of 23 wild type lost ESCA like this, but only 3 out of 18. And in other words, 15 out of 18 had kind of looking intact skin. And once again, this phenotype was reversed or corrected by adding pretreating MBL no mice with recombinant human MBL. And again, C3 complement 3 or complement 4, complement 1Q knockout mice were more like a comparable to white type mice. And we wanted to see what's going on. So we did histological analysis and kind of adjacent skin to the burn area. And one of which we did was to look at MMP. We used fluorogenic substrate for collagenase and gelatinase. You can see this green area. Those are MMP active, while we didn't really see much in MBL no mice. And more surprising was MBL no mice, the skin got almost like a 2 or 3 times thicker overnight. And we didn't really do anything further. Now, many of these experiments were done using anywhere between 8 and 12 weeks mice. Now, we wanted to know if there's any age-related issue. So we aged them average like 20 months. And what we saw was nausea, non-alcohol liver steatohepatitis, or simply fatty liver. And when we sent organs to histology, we sent out. And one of the things they noticed was fatty change, meaning the color of liver was actually different. And so we looked at, we did oil red or staining, which stains lipid. So this is wild type and 20 months old. You can see here and there this orange color that means the lipid droplets. In contrast, we saw two types of lipid deposit, in small deposits all over, or a huge lipid oil red or staining. And we consulted pathology, and they said they see similar pattern in the human as well. And we also looked at fatty acid analysis, and 20 out of 23, I think 16 and 18, we had only 3 each, white type and MBL knockout mice, but 16 and 18 change seems to be higher in MBL knockout. Now, we never published it. We couldn't find any collaborator nearby. So, but we noticed the MBL no mice are tame and very sensitive to any stress. I know this characteristic is the most relevant to this meeting, but we haven't really done anything. The one thing maybe close or at least something happened in the brain is one collaborator at the time was doing traumatic brain injury in mice. So Mbl mice had a slow recovery. So, this was just only in vitro. Lastly, this probably relate to brain house cleaning. So this was just like an ELISA type simple binding assay, but amyloid beta 40, 42, and this is just as a kind of control for the experiment. And red crossed bars shows you their binding as 100%. And interestingly, the sulfate sugars inhibited the binding to A beta. And based on this results, even though people didn't really think this system-rich domain as a binding function, it's most likely this system-rich domain, not just bundle, make form oligomer, but functioning as a binding domain. And so I'd like to believe that Mbl probably working to maintain balance of amyloid beta. Now we know brain seems to need amyloid beta to brain health, I believe. So I think somehow Mbl participate sort of orchestrating whole innate immune or homeostasis somehow. And so the summary of Mbl effects. So the Mbl status is unnoticeable in general. You wouldn't know, you don't feel it, but it might become an issue if you have underlying conditions and some infections. And can be 3 ways. Of course, you know, just healthy, nothing, but too little or even too much seems to be not really good. And when I started, I showed you earlier, we had only a few clinical observations, but now the list has been expanding, and autoimmune diseases and also Crohn's disease and some neuronal programs. And also when we studied, only deficiency was kind of big deal, but now we know too much is not really good either. Now, I'd like to touch a bit what I'm primarily involved with is the development of PET tracer. To me, seeing is believing, and but pain-free kind of slow development for PET tracer actually. Now, one of which successful is tracer is developed by Dr. Brugaloras, and it's on a phase 1 clinical trial, which is 3F4AP. It's a derivative of MS drug for amylopyridine. So this is a cation of a neuron. Neuron is covered or protected by myelin sheath. But if there is any damage, including MS, could be stroke or even Alzheimer, this myelin sheath is damaged and exposed potassium channel, voltage gated potassium channel became exposed, and this tracer can bind. And so this is one, this is, yeah, published. So the, sorry. So we were lucky, I'm sorry, but monkey had an accidental brain trauma injury at this area. And compared to other sort of commonly used tracer, PBR28 or PIB or FDG, 3F4AP picked really more strongly. And so the currently under production studies, these tracers are made by radiochemists, and metabolic related receptors and some inflammatory related molecules, and also pain. And we hope that the tracer, if we hit the right tracer, we could provide early non-invasive and objective diagnostics, and possibly therapy too, depends on what we are doing. And lastly, the acknowledgement, I'm member of the Gordon Center for Medical Imaging, and particularly thank you to George L. Fakri, he's the director, and my collaborators, Pedro Burgos and Norman Dean, and many people are sort of helped me to study Mbl project, and Alain Zuckerts, who cloned Mbl both human and mouse. If I didn't meet him, I would not be working on Mbl, and Mike Super and so forth. Thank you very much.
And thank you, Kazu. We are short on time, not your fault, because you also inherited a little bit of a delay there as well. But what I think we should do is probably move to break until 11:25. So it's a short 7-minute break. I think we all need a little bit time to stretch our legs. But I would encourage everyone to put your questions for Kazu in the Q&A. I know I have a question, which I'll enter into the Q&A. That way, at least we'll both be able to have a break. And